Hello and welcome to this lesson for Advanced Higher Biology, which today is looking at the generation of a nerve impulse in multicellular organisms. So previous to this, we have been looking at the need for communication in multicellular organisms, the fact that different cells are responsible for participating in different functions, and that in order to control all those functions, these cells need to be able to communicate with each other. We've looked at the both types of signalling molecule. We've looked at hydrophobic and hydrophilic signalling molecules, and what we're going to look at now is what can then happen as a result of some of those signals, and we're going to look at generating a nerve impulse. So what you're required to be able to do is state what is meant by the resting membrane potential and how this can be changed in order for a nerve impulse to be transmitted. You need to be able to state how neurotransmitters initiate that impulse, explain what is meant by the depolarization of the plasma membrane and describe the effect that this has, and explain how the inactivation of sodium channels and opening of potassium channels restores the resting membrane potential. So to begin, we'll just have a quick recap of what we mean by neurons and neurotransmitters. So those of you who did higher human last year will be a little bit more familiar with this than those of you who just did higher. Essentially, a neuron is a specialised cell which is responsible for carrying nervous impulses around the body. So they receive an impulse and translate this into an electrical signal which goes across the axon of the neuron to the terminal, which will then create some kind of response, often going to another neuron. Most of this communication between the different neurons and to stimulate the response comes from something called neurotransmitters. So neurotransmitters are chemical messengers, and what they do is they travel across the end sections of our neuron. So if this is the end of one neuron and the beginning of one neuron, okay? We've got our neurotransmitters are being held in vesicles at the ends of one of our neurons. They're stimulated to be opened, and those neurotransmitters are going to travel across something called the synapse, which is the gap in between the neurons, this little section here. So the neurotransmitters travel across the gap, this synapse, and reach the postsynaptic membrane, which is the neuron on the other side of the synapse. And when they do that, they're going to reach the receptor cells, and those neurotransmitters are going to bind to the receptor cells and create some kind of response. And it's this response that we're going to be looking at today. Essentially, our neurotransmitters are going to be able to initiate a response in the second neuron, the neuron that's following on, by binding to the receptors at the synapse, at this section here. So if this is our original neuron which has received some kind of signal, it's translated that into an electrical signal, transmitted it all the way along the axon. We've gotten to the end, we've got these synaptic vesicles, and all that means is a vesicle that's present at the synapse. We've got our synapse here in the middle, which is the gap between the two neurons, and these vesicles are then binding with the membrane and releasing the neurotransmitter. So the neurotransmitter diffuses across the synapse into the dendrite, which is the starting section of the next neuron. And when they reach that, they're reaching these receptor molecules which are present. And they're going to be able to bind with these neurotransmitters. So neurotransmitter receptors are ligand-gated ion channels. So we've looked at those already. So in this case, our neurotransmitter is acting as a ligand, and it's causing a conformational change in this channel. And when it causes that conformational change in the channel, that opens it up, and that's going to lead to a cascade that we're going to be looking at in a second. So the other thing we need to remember about for this section is what we call resting membrane potential. So a membrane potential is the electrical potential difference created when there's a difference in the electrical charge on the different sides of the membrane. So here we've got extracellular fluid is positive, and in the cytoplasm, the intracellular fluid, we've got an overall negative charge. So in neurons, our resting membrane potential is typically between minus 60 and minus 80 millivolts. When we transmit a nerve impulse, what we're looking at doing is changing the membrane potential of the plasma membrane of that neuron. So if this change in membrane potential is big enough, it can trigger what's referred to as an action potential. And that action potential is a wave of electrical excitation along a neuron's plasma membrane. So when that neurotransmitter binds to its ligand-gated ion channel, that allows the entry of positive charged ions, and that begins this change in the plasma membrane, which then allows it to affect other parts of the plasma membrane, which results in this overall wave of electrical excitation along the neuron to get to the other end. So we're going to look in a little bit more detail about how these ions are doing that. So begin with our membrane in its resting potential, and that means there is a higher concentration of sodium ions, which are the green ones, and outside the cell, and a higher concentration of potassium ions inside the cell, and those are our blue ones. And this is giving us an overall positive charge outside and an overall negative charge inside. And this gives us our resting potential of around minus 70 millivolts. This is referred to as polarization because we've got one charge on one side and another charge on the other side. So when our neurotransmitter binds to the receptor at the synapse, that receptor's our ligand-gated ion channel causes some kind of ion movement. So it's starting to let some ions into the membrane. If we wind up with sufficient ion movement, that triggers the opening of voltage-gated sodium channels. So here our sodium channels have opened up, and we're starting to see the movement of our sodium ions going down their electrochemical gradient, and that leads to a rapid and a large change in the membrane potential. So the interior of the membrane now has a positive charge, and the exterior of the membrane now has a negative charge, and that's referred to as depolarization. When this positive charge reaches a maximum voltage, we have our sodium channels becoming inactivated. So they've closed off. Our voltage-gated potassium channels, on the other hand, are then able to open, and that allows our potassium ions to move out of the cell to begin to restore the resting membrane potential. And that's what's referred to as repolarization. So movement of all these potassium ions back out, we're returning this inner section of the membrane to a negative charge, and the exterior of the membrane is now more positive. So our potassiums have moved out to create that happening, and that's referred to as repolarization. Eventually, our potassium channels are also going to close, and the sodium-potassium pump, which is this section here, is going to work to transport the excess ions back in and out of the cell. So it does the last final little tweaks to return the membrane back to its resting potential. That also allows our sodium channels to return to the appropriate conformation. So they return to the original conformation, and that allows them to respond again. So they can, if there is another depolarization of the membrane, if another neurotransmitter binds, they're able to then open again. They're in the correct conformation to be able to do that. You may also see this written as an action potential graph, and that describes the changes of the membrane in millivolts against time. So you can see here we've got our measuring our millivolts. We've got 40 millivolts is approximately our highest, and this would be our zero line here, and we've got minus 70 millivolts, which is our average kind of lowest. And to begin with, we have our membrane at its resting potential of around minus 70 millivolts. So to begin the process, we have our neurotransmitter binding with its receptor protein, and that leads to it opening up those ligand-gated ion channel that is its receptor protein and leading to some ion movement. When this ion movement reaches a threshold value, that stimulates sodium channels in the protein to open, and that causes sodium ions to diffuse in, resulting in depolarization of the membrane. Shortly after opening, the sodium channels become inactivated, and we then have voltage-gated potassium channels opening to allow the potassium ions to move out of the cell, and that helps us repolarize the membrane. When the potassium ions close, we then have the sodium potassium pump working to restore the membrane back to its resting potential. So when this is happening within the plasma membrane of our axon, so our initial response has started here and it started to travel along to get to our axon terminal. Essentially what's happening is when we depolarize one patch of membrane, that causes the neighbouring regions to become depolarized and go through the same cycle as the adjacent voltage gated sodium channels are opened. So essentially, if we're looking at this section of plasma membrane, if it's been stimulated and we've gone through this cycle of polarization and repolarization, that's caused the next section to do the same thing, causes the next section to do the same thing, and so on, and so on, and so on, and so on, until we're managing to move this action potential all the way along the membrane. So if we were to look at the overall structure of the membrane, you would see an area of depolarization along with the following areas resting. Then you'd be able to see the next section would also be depolarized. The section previous that would now be repolarizing, and the section next would still be resting. And then again, this one would be returned back to resting, this one would be repolarizing, and this one would be depolarized. So we'd be moving along the axon in this direction and allowing the signal to reach the axon terminal. When it does reach our axon terminal, what's happening is in this section here, we've had our action potential worked its way along. We've got to the synapse between the two different neurons. What happens is we're going to see that causing those vesicles containing the neurotransmitters being stimulated to bind with the plasma membrane, release the neurotransmitter, and the neurotransmitter is going to stimulate the response in the receiving cell, the following cell. So just like the picture we looked at the very beginning, you saw those neurotransmitters being able to come across that synapse and stimulate response in the next cell. So there you go, that is the transmission and generation of a nerve impulse. I appreciate some of that's a little bit complicated, and please feel free to ask any questions as we go over this in class next week.